Hello, my name is Ed Chapman, and this video cast is going to explain how gel electrophoresis works and why scientists use this as one of the big workhorses or main tools for biochemistry and DNA technology lab work. Okay, gel electrophoresis is a tool used to sort DNA fragments by size. Um, what you get at the end of gel electrophoresis is something they call a gel that contains a barcode-like pattern. So it's very easy to read differences between the different patterns on the gel. This tool works because restriction enzymes cut DNA in a very consistent way. So if you have two samples of DNA and they're identical, they're going to contain identical restriction sites. And that means when you treat them with a restriction enzyme, you're going to end up with an identical set of fragments. The other thing that makes it work is that DNA is a negatively charged molecule. And so if you expose it to an electric field, it's going to move away from the negative pole towards the positive pole. Now, your first step in doing gel electrophoresis is to get your DNA sample, and this usually involves lots and lots of purification steps. And you may also end up with just a very small amount of DNA that you want to amplify. And so, of course, the easiest way to amplify DNA is to use polymerase chain reaction. So at the end of that process, you end up with lots and lots of identical clones of your original piece of DNA. Step 2 is to fragment your DNA using a restriction enzyme. And remember, restriction enzymes work because DNA code contains certain sequences or restriction sites at which the enzyme can cut. So once you mix in the enzyme, it will cut at all these restriction sites and fragment your DNA. Step 3 is you're going to add your DNA fragments to a test tube or to a little vial, and you're going to add a dye to it to make the DNA show up. So you just need a chemical basically that will stick to DNA and make it show up, because DNA is pretty colorless on its own. The dyes that are used for this purpose are almost always fluorescent, and they show up really well under black light. Step 4 is you're going to load the DNA into your actual gel. Now in this image, you can see that the pen being held, or the little injector being held by the lab tech, is being used to carefully add the liquid DNA, which is blue in color, to these little depressions or wells that have been cut out of a piece of gel. The gel can be made from a number of different materials depending on the size of the fragments that you're working with. The gel is submerged in a liquid, so you literally have to stick the tip of that little applicator pen underneath the liquid and carefully add the blue DNA containing liquid, which is much denser than the buffer that the gel is soaking in, so it will sink into the well and stay put, even though it's a liquid in a liquid. Notice that there are multiple wells or multiple depressions in the gel. This is because when you turn on the electricity, you want to compare the pattern of bars of fragments, and they have to be beside each other, so that in a way, you can think of it as an experiment with all the constants. So each little well or each little sample of DNA is exposed to exactly the same conditions, so they produce a readable result. Your fifth step is you're going to actually expose this gel. Here we're looking down at the top of it in this diagram to an electrical field. And remember, DNA has a negative charge, so it's going to move away from where you put it towards the positive pole at the opposite end of the rectangle of gel. And so after half an hour to a couple hours, the DNA will have separated into visible bands according to length. And you leave the power running until your bands approach the opposite end, or the positive pole. If you let the electricity run too long, all your little bands will run right off the other end of the gel, and you won't be able to read it. This works because the longer the fragment, the slower it's going to be pulled through the gel. So the longer fragments of DNA are going to be close to where they started, up where the wells are, and the shorter fragments are going to be closer to the positive pole. And step 6, finally, is to look at your gel and compare your lanes, or the strips of bars that come from each sample of DNA that you started from. And in this particular diagram, you can see that all 1, 2, 3, 4, 5 of the DNA samples are different because each one has a slightly different banding pattern. Thanks for listening. We will stop there.